It is a very great pleasure to introduce the 2009 David Marsden lecturer, Professor Rick Morimoto. The David Marsden Award Lecture recognises exceptional contributions to the neurobiology of movement disorders. Professor Morimoto is recognised for his work on the heat-shock stress response and the function of molecular chaperones, and the title of his lecture is The Stress of Misfolded Proteins in Aging and Neurodegenerative Diseases.
Well, first I'd like to thank the organizers for the distinction of the David Marsden Lectureship. What I'd like to do in my talk today is to try to bring together a lot of different ideas and hypotheses at a very basic science level to understand the role of protein damage and the importance of protecting the proteome in many basic science and disease states. First, I think, is the very important observations over the past 5 to 10 years that it's a large number, nearly 300 different human diseases, which are all due to the appearance of misfolded and damaged proteins. And so as you look at this list, you'll find different states, metabolism, the immune system, transport, or the nervous system, and you'll see different proteins. For each of these proteins, it's been well demonstrated that mutations in these proteins lead to their instability, that misfolding occurs, that aggregates and occlusions appear, and that this seems to be an important base of toxicity. In fact, in Dr. Lang's talk that we just heard, we saw evidence of these aggregates of tau and other proteins. The fundamental behind this, literally your 1 minute of biochemistry 101 who might have forgotten it, is that all polypeptides share in common, regardless of the species of the organism, that you start off as an unfolded polypeptide here, you then make transitions to intermediates, then you form the native polypeptides. These polypeptides then turn over through a normal clearance mechanism. And when the proteome is in a stable state, all proteins behave. The problem is the environment or mutations now alters and destabilizes these intermediates, leading to their appearance of misfolded species. One profound example is with cystic fibrosis, where the deletion of a single amino acid, delta F508, leads then to the premature clearance of an essential protein in the lung alveolar cells, leading to CF. Another example is with alpha-1 antitrypsin, where the mutation now leads to improper trafficking, and the result is, of course, emphysema. What I think all of us are brought together by are these diseases of toxic folds, as reflected by Parkinson's disease and Parkinsonism, Huntington's disease, ALS, and Alzheimer's disease. And in each of these cases, these toxic species are a direct consequence of the accumulation of misfolded and damaged proteins. And we know that there's a class of proteins that are as ancient as all of biology. You know them as molecular chaperones or heat shock proteins. These proteins are essential to capture proteins as they misfold and to prevent them from being damaged. In doing so, what the chaperones do is they facilitate the formation of the native state. So the emphasis is really on misfolding and this idea of proteostasis, that there's a large network of genetic interactions that if we understood them well, we could apply that knowledge and develop a therapeutics to actually prevent misfolding from happening. What I'm showing you here is a little bit more complicated slide, and what I want you to pay attention to is you'll notice that we start with DNA, going to RNA, going to proteins. And that's, of course, fundamental biology. But it's been assumed that once a protein's been made, that's the end of the discussion. The problem is many proteins, alpha-synuclein, Huntington, A-beta, rapidly form aggregates, which you can see down here. And the problem is you get competition between the formation of a normal protein, the appearance of aggregates, biological degradation, and biological misfolding. In other words, it's not easy being a protein. It has too many choices, and many of these choices are very deleterious to its life. You notice that for every one of these reactions, we have a forward reaction and a backward reaction. What this means is we can start to understand the underlying biochemical and biophysical processes that lead to the stability of individual proteins. This, as you'll see at the end of my talk, gives us an opportunity for therapeutic intervention. So proteostasis then is the biology that protects the proteome. And we'll talk about how these disease-associated aggregation-prone proteins interfere with the stability of many other proteins in the same cell. And moreover, that the real challenge is trying to understand how our proteome, how our cells deal with stress and aging. These seem to be the rather profound challenges in biology. And shown at the bottom is sort of this balance, that as misfolded proteins accumulate during aging and are challenged by different disease states, the proteostasis network works hard to restore balance. I think what you're all familiar with, of course, is the story of Huntington's disease. And what you see is a patient in the villages near Lake Maracaibo in Venezuela, where there's 14,000 descendants of a single founder, a total of 18,000 since the mid-1800s. You're all familiar with, I think, the profound degeneration that occurs in the brain of individuals with Huntington disease. And shown over here are just examples that we're all familiar with in familial neurodegenerative diseases, that it's daunting and devastating to families that are so afflicted. We know the molecular basis of these diseases, and shown here, in this particular case, it's an expansion of polyglutamine, where in the normal case of the disease, you have short polyglutamine, such as Q19. In the disease state, it expands. And shown at the bottom, you can actually see the appearance of these polyglutamine aggregates as the length of polyglutamine increases within the cells, the neurons of the affected individual. Perhaps one of the most striking aspects is anticipation. Shown at the top is a table where you can see the length of polyglutamine on average, and you notice that as the length of polyglutamine increases, that individual is increasing at risk as you make the transition across the mid-30s, such that they're not just carriers, but they're children. As the polyglutamine expands in the Huntington protein, now will come down with Huntington's disease. And of course, what's striking and dramatic is the distribution of the CAG repeats in Huntington's and all of the 9 other polyglutamine diseases, the ataxins, the Kennedy's disease. And what we know is that as the polyglutamine length increases, as shown in the bottom in the right curve, that there is an inverse relationship with the earlier onset of the disease, which you can see as the red squares. Now, what's striking for all of these diseases is, despite the genetics, that there are other modifiers. That's shown in the pink in the background is actually that you can have twins with exactly the same length of polyglutamine repeat. Unfortunately, both twins will both succumb to HD, but the course of the disease will vary quite significantly, indicating that there are other modifiers that are critical as to how the polyglutamine affects the cell and the organism. What's been, I think, the most rewarding over the past decade has been, because of the fundamental protein biology and protein misfolding, that one can study this in a test tube. You can model it, you can purify these proteins and study them with great elegance using biophysical methods. And from this, we've learned an amazing commonality. Whether it's synuclein, whether it's Huntington, whether it's A-beta, all these proteins have a propensity to form alternative stable structures. And these structures in vivo, whether they're in tissue culture, in yeast, Saccharomyces cerevisiae, the baker's yeast, C. elegans, Drosophila, or in mice, show nearly identical characteristics. which of course provides us great opportunity that you can then use the genetic model systems in biology to do discovery. Organisms such as yeast or worms or flies, where they live a very short lifespan. And you can do experiments in a period of days and weeks, which in mice would take many years, and for humans we can only extrapolate to. So let me just sort of point out, since most of you are unfamiliar with C. elegans as a model, but actually you're familiar with the fact that over the past 4 years, 5 of the 6 Nobel Prizes went to C. elegans, and there's a simple reason. It was the first genome sequence. It has exactly 959 cells as an adult, of which a third are neurons. And you can see the animal as it passes through. It's transparent, which means that we can introduce genes at will and make fluorescent proteins. It has a short lifespan and a short life cycle. For those who are wondering what we can learn from worms, it's the discovery of cell death that came from C. elegans. The concepts of lineage came entirely from C. elegans. So I think we can learn a lot from these model systems. So what our lab has done over the years is to recognize the opportunities of this biological system, introduce, for example, polyglutamine stretches, as you can see here, tag the polyglutamine with a fluorescent protein from an Atlantic jellyfish. That's what won the Nobel Prize by Marty Chelfie this year. And you can see the advantage. You can see now aggregates as the polyglutamine increases. We've done the same thing with mutant SOD, and you can see the appearance of these very striking yellow aggregates in these animals, and we've done the same thing with A-beta models. And the point is you can do this, and here's an example where we've done this with alpha-synuclein. These studies were done by Ellen Nolan, a former postdoc, where not only can she make these aggregates, but you can see below in the colored images that we can then use biophysical methods to now quantify the characteristics of these aggregates. And the striking thing with alpha-synuclein aggregates is they form 2 different classes. Some are soluble and diffuse, others are inert in the same cell type. Whereas Huntington forms aggregates that are only inert, and SOD forms aggregates that are only soluble. So even though each of these proteins misfold, we can already start to understand the underlying characteristics. Now let me just show you an example of how we can use this as a very powerful tool. You're looking at animals where you can see the Q18. Actually, you can't see it at all. Okay, so unfortunately, under the Q19, there's supposed to be a movie where you can see the animals, and in Q82, you get the aggregates. What you can see is if we take animals that are at an intermediate length of 35 residues, and you notice as the animals age, you get age-dependent aggregation. And you can quantify this readily. And when you quantify this, what you can see is as the polyglutamine length increases, you get an age-dependent increase in aggregate number. And as this occurs, the aggregates are toxic. In other words, here we have a model in a C. elegans, where whether we put it into muscle cells or neurons, we get exactly the same phenotype. The polyglutamine is toxic in real time, and it causes age-dependent aggregation. What this allows then is a series of experiments. Why is this toxic to the cell? What's the basis for late onset proteotoxicity? In other words, the aging question. Could this help us understand cell-specific specificity or toxicity? And what are the genetic modifiers? What are the genes that the organism uses to protect ourselves or are at risk? And finally, what happens when you go from a cell to an organism? So I'm going to summarize a lot of different data from our laboratory. What we've learned is that there's a very delicate genetic interplay. That when you take a dominant folding mutation, such as a polyglutamine or a synuclein or a SOD, it causes misfolding and aggregation, as I showed you. That then directly competes with the folding quality control machinery in the same cell. The consequence is many other proteins become at risk. The rest of the proteome is not as stable as you might have thought. And so when you now start to take away critical folding machinery, many other proteins start to misfold. And the consequence then is an age-dependent amplification of proteotoxicity. What this helps us start to understand is cell specificity, because protein polymorphisms are tissue specific. And it has then conditional effects on the quality control machinery. We've used these approaches to then do large numbers of genetic screens. So we already have an idea how many genes are there that protect the proteome. We can identify proteome protective networks. These are transcription factors such as heat shock factor or DAF16, which is called FOXO3, and these are master regulators of protein stability. We've identified, as you can see, nearly 600 genes. And you might say, isn't that a lot? And that's actually the good news. Turns out that enhancing any one of these 600 genes actually can protect the proteome because it's a highly integrated network. And all of the genes that we identify in C. elegans, over 95% of them are conserved all the way to humans. And many of these have then been subsequently validated. So I'm just going to show you one example, which really came as a surprise. C. elegans was the basis of the discovery of the genes for longevity. The first set of genes that were identified were the insulin signaling pathway, which makes sense. Modulation of insulin signaling, in particular, downregulation of insulin signaling, leads to lifespan enhancement. And what we found was those same genes, when you enhance lifespan, suppress protein misfolding completely. And you can see this in a very simple example. If you look at these animals, you can see that there's a C. elegans and it has a lot of aggregates. If we now shut down the insulin signaling system, you see the protein is now soluble and diffuse. If we then knock out the downstream transcription factor that's activated when you shut down the insulin signaling, you go back to the aggregates. And shown in the bottom panel is literally the shift as you go from toxicity to suppression of toxicity. So what this starts to tell us then is these are the two major activators, heat shock factor and DAF16. These are transcription factors that the cell uses. It's present in every organism that we know of. And these transcription factors sense protein damage and cell stress and tell the cell to protect itself. Now, we can then ask questions, well, what about aging? I gave you a little hint earlier that there had to be something going on with regard to lifespan. And so we've generated animals where we now introduce what are called folding sensors, proteins that are at the cusp of stability. And in this case, we use firefly luciferase. If we put it into worms and now expose them to heat shock or polyglutamine expression or an oxidant, paraquat, you notice that there's a reduction in activity. But what's dramatic is aging. If we just take these worms and now watch what happens to the activity of the enzyme, you'll notice by day 5, which is right about here, there's a 50% reduction in activity, but shown below is the Western blot analysis, which tells us the protein's fine, it has no activity. And the reason why the protein has no activity is it's not properly folded. In other words, aging is a culprit. Now, when is this occurring? This is not mid or late in the lifespan of the animal. It's early in adulthood. So I want to show you a couple of other examples. You're looking at a protein called paramycin, which is an essential protein in muscle cells. And we've used a mutation where it doesn't fold well at the restrictive temperature. That's an elevated temperature. And you can see that if you look at this last panel right here, that all the paramycin looks like aggregates. It's not functioning. But early on, you see that all of the paramycin's properly folded, and you can see the proper muscle striations. And what I think you can all see is by day 3 in the temperature-sensitive mutant, you now see aggregates, which means that the protein is misfolding as the animal ages. And if we now look at 4 other proteins, RAS in neurons, acetylcholine in neurons, GAS1, which is a mitochondrial protein expressed in neurons, or perlecan, a protein expressed in muscle. In every case, these proteins all have what are called missense mutations that affect their stability. But what you notice is they all lose activity during early adulthood, which is telling us then that there's a commonality and the proteome becomes destabilized just as a consequence of aging. So in thinking about this, we asked, well, what could be the basis? And what we discovered is that our essential stress responses that protect ourselves, in other words, how the organism senses the environment and protects the proteome, this is called the heat shock response, is dramatically reduced at day 4 and stays down at day 7. Not only that, but also all the proteins that go to the endoplasmic reticulum and to the Golgi are also at risk because the unfolded protein response is dramatically reduced. And if we then look at other chaperones, such as HSC70, you see there's no change in aging. But what it tells us is that some of the most ancient cytoprotective stress responses that are there to detect protein damage literally abandon us during aging. But you notice it's not down completely. There's still some there. So what we discovered was that we could now enhance it. So shown in this panel, you're looking at RAS, which loses 50% activity at day 5. If we now enhance the transcription factor DAF16 or HSF, you'll notice in the green lines, we can actually prevent the loss of RAS activity, which means RAS as an essential protein in the proteome no longer decays. We can now restore the proteome by selective activation of key transcription factors in the cell. So there's hope. It means we might be able to stabilize and reverse this age-dependent decline, which we think is then the basis for protein misfolding diseases. Now, one of the things we've learned is that these key transcription factors work together, and they work together in interesting ways. And a recent discovery by Anne Brunet was that there's a protein called Sirtuin. Many of you probably have heard about resveratrol. Resveratrol, of course, is a product, a natural product out of red wine and out of grapes and various fruits and vegetables, and you can buy it at any grocery store, and supposedly it protects you forever. Well, one of the reasons why it's sold and so popular is it activates supposedly a protein called Sirtuin. Sirtuin is an enzyme that affects the activities of key proteins in the cell. What I'm going to show you is that Sirtuin protects and enhances the proteome directly. So it provides a direct basis for caloric restriction as protective and lifespan enhancement. So in order to do this, I just want to give you a sense of how our cells see protein damage. In a normal cell, you have native protein shown in blue, and you have heat shock factor present in the same compartment. When protein damage such as alpha-synuclein or Huntington or A-beta appear, it increases the amount of damage in the cell. The transcription factor HSF now senses this, and it shifts in equilibrium so that these chaperones, these little sort of hexagonic structures, now bind to the damaged proteins to prevent their further misfolding. The heat shock factor now becomes activated. It goes into the nucleus, binds to the heat shock elements upstream of the chaperone genes. You increase the expression of these hexagon chaperones to bind all the misfolded proteins. This happens literally within seconds when protein stress and damage is sensed in the cell. When the stress is removed, the proteins are folded, our cells return back to normal. What we've learned is that the role for Sirtuin is at a very specific step. It plays a role in whether the transcription factor is bound to DNA or releases, and what Sirtuin does is it directly regulates its binding. So what this tells us then is that the way our body, our cells see stress is that this transcription factor sees it in 3 different ways, the most recent one being that it directly keeps the factor bound to the DNA, keeps the chaperone levels high to protect our cells. And this happens when you reduce the caloric intake or you activate the system. So what this tells us then, in a very interesting way, is that very important transcription factors, P53, which regulates the genome, NF-kappa B, which regulates the inflammatory response, heat shock factor that protects the proteome, and FOXO that regulates stress responses, are all tied into this single protein, Sirtuin. And when Sirtuin is activated by resveratrol or caloric restriction, you now put the cell and organism into a protective state. It senses damage more quickly. It now resists the accumulation of damage. It prevents apoptosis and influences lifespan. So what I've told you in this talk is how we've thought about this idea of the cellular challenge and cellular toxicity, the importance then of these chaperones and stress responses, and that aging really is the centerpiece at the molecular and cellular level. By understanding what happens during aging, we will understand then age-related degenerative diseases. These two go hand in hand. Now, I want to sort of give you a sense that there's more surprises. A lot of biology is done with yeast. And we know that almost everything we have is conserved in yeast. But yet, are all stress responses conserved in yeast and worms and mice and humans as well? And I want to show you a little result just to give you a sense of an exciting future. In C. elegans and likely other metazoans, they sense the environment through what are called sensory neurons. In C. elegans, there's two. They're called the AFD neurons. These genes I've listed are genes expressed only in these neurons. And what happens is if you knock them out, what's shown here is that the activation of heat shock genes in the remaining 957 cells is shut off. In other words, the neurons sense the stress, integrate the environment, and tell every other cell whether to protect the proteome or not. This is exciting because it offers a very different way of thinking about how you might protect an organism from protein damage. And just to show you how striking this is, here's a worm that we've engineered with a reporter for the heat shock response and becomes fluorescent. When you now are missing these sensory neurons, just two, the heat shock response is reduced dramatically. So this really gives us a sense of a new way of thinking of how the organism integrates stress from nutrition and neuronal signals. I want to close this talk by giving you a sense then of how we're thinking about the future. Protein damage, oxidative damage, and other forms of damage really impinge on the stability of our proteomes. It's a systems biology of proteostasis, transcription factors that are important, and all integrated to stress biology, nutrition state, and health span. What I've told you about is that as cells and organisms age, there's a collapse of protein homeostasis, and as this occurs, cellular function decays. Our protein homeostasis machines that I've described to you are protective. They determine where on the x-axis the cell succumbs. The organism becomes at risk. As the organism expresses these chronically damaged proteins over a lifespan, it leads to an imbalance. And the goal is to restore proteostasis. Can we do this through therapeutics? And it turns out we can. So what I'm showing you is a complicated diagram. It's a network. Each one of these different colors correspond to activities in the network. And above that correspond to different gene switches. You'll notice DAF16 and HSF are right there. These are gene switches that we think we can control to protect the proteome. And you have different pathways coming in from the unfolded protein response, the proteasome for clearance, calcium signaling, or oxidative damage. What our laboratory has done is finished a 1,000,000 molecule small molecule screen to identify a whole new set of proteostasis regulators that enhance these molecular chaperones to protect cells from the onslaught of damaged proteins. And what we think this is going to allow is these proteostasis regulators will recognize the imbalance network and by therapeutics now restore it to prevent protein misfolding disease from persisting. So I'd like to thank everyone in this audience the members of my laboratory, my collaborators worldwide, and my funding sources, and again, the Movement Disorder Society and the recognition of the David Marsden Lecture. Thank you.